All right. Let's see. Okay. That seems to be live. I was about to go to bed. I'm unsubbing. That's hilarious. 1 second, bro. 1 second. Give me a second, guys. Give me a second. I'm getting stuff put together here. When the night, I got this damn song stuck, man. And the land is dark, and that moon is the only light you see. Yeah, we got the chat on the screen. Let's go. Oh my God, it's all coming together. What's up, Big Steve? Standing by me. Talent off from the bath. Nah. That was earlier. We're good. So darling, standing by me. What's up, beautiful people? Oh my, yo, he's back? Yo, and my takes is back. Let's go. Come on. Let's go. Why are you booing? Wait, yo, stop throwing stuff. Come on. I can't just get on live stream. Really? I thought I'd at least. Okay, Captain, you miss me, I'm sure, right? Don't ask me about cross-contaminating the microbes, Captain. Never mind. Okay, you know what? Forget about it. Well, I'm here. All right. Whether or not I'm here or not, I'm here. And yo, there's gonna be some shilling involved in this stream. Captain's Critters gave me the hookup. Man, I turned this tank green in 2 days with the help of Captain's Critters. You're looking at a 10-gallon. It's solidly green. Thank you, Captain, for ruining the aesthetic and viewing capabilities of my tank. It's exactly what I needed, and I put the Daphnia right in there. They're going crazy. They love it. They absolutely love it. So, you know, if that doesn't go well, I have something to dump on someone like it's the Nickelodeon slime. I'm just gonna take the 10, go to the next time. You know what? Next time someone else wins Breeder of the Year award, you know, I'm gonna bring my green water with the Captain's Critter logo on the front, and I'm gonna pour it straight on to whoever beat me. But what is up, people? How's it going? There's a lot to catch up on because technically I didn't even do an auction review. If you guys want, we could do an auction review before I do the Daphnia stuff, if you want, or I can do it afterwards. You know what I mean? But I figured it's been a while since we learned anything on here, so I figured we may as well get into the Daphnia. I just got some Daphnia, so you know it was kind of like good time. You know, I was reading about it anyway for my own personal benefit, so you know it's kind of like let's do a stream. You know what I mean? What's up, JP? What's up, Mick? Adam, outside in, how's it going? My friend, my guy, Big Steve, the hottest up-and-coming hobbyist breeder and best breeder of Nanochromis transvestitis in the world. Big Steve, glass boxes. What's up, man? Yeah, we got the right people in the building today. I saw everyone in the chat. Sutton, what's up, Sutton? A couple of fish. Johnny Best, how's it going? JP says, deep dives. Captain Curtis said, not the cross-contaminated Warren tubs. Yeah, I doubt they're cross. I doubt it. We're good. We should be good. We should be good. I don't use any paintbrushes. I use I either use a spatula to do microworms or I use aquarium tweezers because it's like straight. So I just scrape it off the side. Sage K's, how's it going? So if you guys didn't know, I've been loading up on my worm cultures. So I got microworms here. That's all worms on the side. And like some of you guys may not know this about me. And Captain, she's a elitist. She don't really respect my game, but like I came into the game with some microworm skills. Okay, so this is kind of like me getting back to my roots because that was the one thing that I was able to do really well at early on. And it's been a minute. So I got microworms. Then I got these are my banana worms. Okay, so this is after like, have I had these a week or so? I don't know. And I didn't even set them up. They were left in the bag for a few days. Oh yeah, the vinegar eels on oats are doing really well actually. And then I got Walter worms too, but they're underneath a bunch of stuff, so I'm not even gonna. You guys get the point. I got the worms on deck. Okay, not inside me, but it is what it is. Steve said I do have a fresh batch of nanochromas. Of course you do. So yeah, today we will get into Daphnia. All about Daphnia. I mean, how many questions could you possibly have about Daphnia? Well, hopefully we'll answer most of them during the stream. I did make a list. I made a list of different sources, different things to go over for the stream tonight. And I couldn't format it right, so I just put it in a Google Drive document. So I'm going to put the link to that Google Drive document in the chat now if you want to look at the possible list of things that I'm going to be going over. And then, you know, I'm going to get to the actual thing. And then again, we probably do an auction review of the last auction I participated in. We'll do a seller's report and see what some stuff went for. But okay. With that being said, any other notes? Captain's Critter says penmanship has left the chat. Yo, no one's got to read the worm containers except for me. I don't understand. I don't understand what the problem is. Why is everyone's a critic? You know what I mean? Everyone's a critic, and no one just appreciates me. That's how I feel. Yo, every time I really start getting to cooking, that's when something with the audio wants to go down. I don't even know what I did. Hold up. Give me 1 second. I think we're back. Yeah, we're back. Anyways, as I was saying, holy, that's right. Yo, Steve, thank you for the membership, Steve, saying no audio. All right, Steve, we're back. We're back with audio. And basically what I was saying was, look, they never appreciate you until they really need something. And until recently, oh, baby brine shrimp, like, we're just going to do what's convenient, what we've always known. But now that the environment's come into play, you know, it's a catastrophic situation out there. Everything's getting, I don't know, hurricaned, flooded, Droughted. You never know what's gonna happen these days. It'll be like, oh, record-breaking disaster. Everything's dead. It's just another day, another thing that we have to deal with. Well, now it's Artemia cyst shortages, and suddenly, oh, Mr. Daphnia, oh, I didn't see you. Oh, fairy shrimp, what do you mean you existed this whole time? What's up, man? Long time no see. Like, we really need to reconnect. 186 said, I once dated a girl named Daphnia. Holy, yo, make sure she on that birth control. That's just, you'll see how we get into this. The reproductive rate of Daphnia is crazy. I'm not taking any chances. Names have power. You know what I'm saying? Nope. Nope. All right. Where did my chat go? There we go. Was she friends with Velma? Oh, Daphne. Oh, Scooby-Doo. We got some people of culture in the chat. Now this is excellent. Okay. my voice is back. We're here. So anyways, it's become a lot more studied recently, which is fantastic for our purposes, because it means that there papers such as this, Daphnia as a versatile model system in ecology and evolution. Every time I hear a model system, I get excited because I'm like, great, they're going to actually detail and explain a bunch of stuff in one place, and I'm not going to have to go like scrape it between 3 different sources. You know what I mean? I'm going to try and stay atop my new best practice where I pin the link to whatever I'm reading at the time that I'm reading it in the chat. So I pinned that. We'll see if I stay on top of this. And yeah, the isopod agenda. So Daphnia, you might have heard them called water fleas. They've been looked at as a ecological model. It's been studied as ecological model for a while. And this paper describes them as a planktonic crustacean with a cyclic parthenogenic life cycle. Okay, they have a nearly worldwide distribution. They inhabit standing, fresh, and brackish water bodies, from small temporary pools to large lakes. Now, I named the stream Daphnia magna because I'm going to try and focus on a specific species, but, you know, let's get the big picture of what Daphnia are in general. Even though I have a favorite, Daphnia magna is what I have. That's what I was looking up. You know what I mean? So, listen, I'm a little bit self-centered. If you haven't noticed. Mick says Springerville Ag is a huge bank of info. Indeed, isopod in the chat. So Daphnia, they're predominantly asexual reproduction, okay? And because of that, it allows for the study of phenotypes, excluding genetic variation, and enables them to separate genetic from non-genetic effects. Fantastic. I don't care about that. I want my fish to eat them. You know what I'm saying? So let's look at the natural habitat and life cycle as described by this paper, okay? So Daphnia is a genus of small planktonic crustaceans with a very wide geographic distribution. Its English name, water flea, includes other members of the Chlorocera order within the class Branchiopoda, united by the morphology of their trunk limbs. There are over 100 described Daphnia species. Wait, Nick said he dated a girl named Daphnia too? Yo, can you just not make everything in your life about the whole fish hobby thing? Like, even the chicks you're dating are named after live foods. Like, don't you feel like this is taking it a bit far? Like, don't you think this is taking it a little bit far? I'm just asking. Okay. There are over 100 described Daphnia species. So yeah, we're not going over 100 species. Let's stick to one. Each having a similar body architecture characterized by a relatively large head. Okay, see? Some of us as I read, you might be like, I have that too. You know what I mean? Characterized by a relatively large head with one simple compound eye and a body encased in a bivalve-like shell. Daphnia are more or less transparent so as to evade visually hunting predators. One thing I've learned, or I've been struggling to try and understand, is how much some fish rely on vision for hunting. And I think the last time we talked about that was when we mentioned how some fry are super small, and that's part of how they can actually survive when they're fries, because they're so small and hard to see. Anyways, more or less transparent to evade visually hunting predators, e.g., planktivorous fish. They molt 4 to 6 times before reaching maturity, but continue to molt and grow in regular intervals throughout their life. Now, as I'm getting into the Daphnia thing, I definitely noticed the molting because when I put them into a temporary like Tupperware situation after I got them, the molts were all over the ground. I was like, "Are the Daphnia dead?" And I took a closer look. I'm like, "Damn, bro, they're molting every day. There's more little molts on the ground. So just as fast as they reproduce, you know what I mean? Each cycle they gotta molt to continue the growth. Okay, that infinite growth. I feel like Daphnia would make really great CEOs. Honestly, they understand how to grow exponentially. Anyways, newborn Daphnia resemble adults, except they lack the well-developed dorsal brood pouch of adult females or secondary sexual traits of males. Captain said those ladies were probably sisters. We got some sister wives situation, and I just put it to live chat mode. Thanks you, Captain, for that. Thank you, Captain, for that. Mark Snodgrass said I currently have 3 tanks of Magna and 2 of Moina. I feed them live green algae. Well, we actually have a live feed of my green tank from the side here that I made green. I've now seeded with Daphnia Magna, and we'll see. You might actually see in the bottom, you can see there is some Daphnia going around. It's only been a week, but there's quite a lot. I actually already fed out there. I netted some out. Anyways, I'm gonna make this slightly bigger, and maybe we'll see them slide through. So here in this little handy dandy, and I'm gonna adjust the dimensions of my page here. That's crazy about the molting. The molts probably make some good shrimp. Yeah, you know what, man? Molts, everything can be reused, right? Everything's a food to something. You know what I mean? Everything's a food to something. Lots of molts though that I've been noticing. So here's the schematic phylogenetic tree of animals with a focus on the Daphnia in the red. Okay, and we see Artemia down here. We got sponges, corals, all sorts of things going on. And then here's the Daphnia in this little tree here in Branchiopoda. So with the Daphnia with Triops and with Artemia in here, and outside around that we got some Melocostraca, Crepedia, Copepoda. I guess is Moina out there in Copepoda? Are they not in here with the Daphnia? I guess we'll have to see. But Daphnia or Claudicera that form together with Nostrasa and Anstraca, the Branchiopoda, formerly sometimes referred to as lower crustaceans. Jesus, we got class wars with crustaceans too? My God. Let's see. Daphnia may be closer related to the model organism Drosophila than to a lobster. Okay, I'm glad that we know that. Here we have some close-up photos of the Daphnia here. 3 Daphnia species often used in biological research, representing 3 major clades of Daphnia within this large genus. So A, we have a baby Daphnia magna. And as mentioned before, you know, the babies do reference or look like the adults. They're just missing the brood pouch that you see here. And if you ever seen a real, Daphnia magna can get pretty big if they live long enough. But even before they reach that max size, you can clearly see the ones that have eggs in them. You know what I mean? Mark said, once a week to feed my small fish, I use a kitchen strainer and put the adults back in a clean tank. Now that's the nice thing about stuff when you get the stuff like Daphnia. Even if you had a brine shrimp colony running, the ability to pick the size by just using a net and like straining it out is kind of nice. Ooh, I just saw isopod go by in the green water. It's crazy. Okay, so this is Daphnia magna up here. And then C is Longispina and D is Pollux. Okay, and it says Magna and Pollux are predominantly pond-dwelling species, while D, Longispina, and related species are often found in large lakes. The 3 adult females carry parthenogenic eggs in their brood chambers. All animals are oriented with their head to the top and the ventral side to the right. The black round object in the head is the single complex eye. The large second antenna are used for swimming. So, swimming with your antennas is a choice. Or is it? I don't know. But I will say they might need to get one of those, I don't know, those things people chew to try and get their jawline going. Like, their jaw's kind of like being enveloped by the rest of their body. I don't know. They might need to start doing some mewing. Is that what it's called? Like, can we get the Chad Daphnia out here somewhere, show them how it's done? I don't know. This guy's got some separation, but the rest of them, I'm not so sure. Also, it says single complex eye. They only have one eye? Really? It's kind of weird. Captain says Daphnia, Pulex needle tail makes them a nope for me. Magna and Moina are around and good eats. Okay. Here in this figure, we have, let's see, we got Pulex with a sexual resting stage. The black structure. B, D Magna with the resting stage, the dark mass in the center part of the body. Also visible in the animal. In A is the next asexual clutch being produced in the ovaries. And in C, we have a male D Magna. And in D, we have freshly cast stage, resting stage from D Magna containing 1 or 2 embryos in development or rest. The resting stage has still parts of the female's carapace attached to it. All right, so yeah, this definitely looks a lot different than when they have the active, ready to go eggs going on in here. It kind of looks, it looks like they have like internal hemorrhaging or something like that. It don't look quite right. 1 second. There we go. There we go. Get my dimensions right. All right. Now this is, what one is the male? C is the male? Interesting. Can you guys tell? I can't. Mark Snodgrass said, my problem is they become a pet and feeding them with fresh algae daily fun, not a chore. Oh, that's very nice. I will say that now that when you have like the green water, like I had the one culture in my tub, and it gets like the thick, it starts settling on the bottom, you get the thick green thing on the bottom. It is very nice. It is very fun to turkey baste her out just solid green into something to feed it. I enjoy that quite a bit. So this is my favorite part, is when they show us some of the natural habitats of some of these things. So here is some of the small pools to large lakes. It says they may colonize saltwater lakes or estuaries, but they do not typically colonize seawater. Some species of related genera do. Daphnia are often a keystone species in ponds and lakes, where their primary consumer, filter feeding small suspended particles in particular unicellular algae. As such, they play an important role in aquatic food webs, being prey themselves for fish or diverse invertebrate predators. Now, this is one thing I like about them as a live food, and why I think stuff like them, well, already is replacing baby brine shrimp for me, is because I do think it's nice and easier for me to feed my fish something that's eating, algae or something like that, than it is for me to try and incorporate the algae into some sort of dry diet. You know what I mean? It's kind of morbid, but in some ways it's easier to feed a live food, the vegetable, than it is to work the vegetable in to like a pellet or something yourself, or to find a pellet that's going to work for you, that stuff will want to eat, that includes that ingredient in the right proportion. Mark says, anyone added cherry shrimp to their Daphnia tanks? Well, there is some shrimp in this green water tank, and there's some aquatic isopods, so I guess that kind of counts, but I just did it, so I can't really tell you how it's going to turn out. All right, so here we see, it says, okay, A is the northernmost located D Magna population so far, located on Vardo Island in Norway. Okay, so this is figure A up here. This is in Norway, the most northern found location of Daphnia magna. Okay, that's interesting. B is a permanent pond in Switzerland. Okay. This looks like a pretty normal, you know what I mean? Normal spot. This stuff always makes me want to find out where the heck this stuff is near me, Outside insect, kind of like gut loading in reptile keeping. Yep, that's precisely. And people do that with, some people do hatch baby brine shrimp and like enhance them with fish oil or something like that because they're not nutritionally complete. But find the right algae, I guess. Who knows? Let's see what C is, because that looks like something temporary, right? A shallow rock pool on the island Grambuskin near Tvermin in southern Finland. Look at this. This looks like it's on top of a mountain or something. I don't know. Like, this looks like a parking lot and it just rained and they haven't fixed the drainage for it in like 10 years. And the concrete's just cracking because every year it gets worse and worse. That's what that reminds me of. Damn parking lot. D is dry rock pool close to the pool in picture. C is the surface is covered with Daphnia resting stages. Ooh, I want to see that. Okay. So it's hard to see even from my point of view, but there's kind of like little speckles on the top of the water. E is a temporary rain pond in the desert in Israel. Wow. Okay. We're getting the international Daphnia tour right now. What's up, Jesse? Captain says, cherry shrimp can't handle the filth conditions most keep their heavily populated Daphnia cultures in, and shrimp tanks generally are too clean for Daphnia to take off in. Alright, we'll have to see how filthy my shrimp in there get. F is a saltwater pond in southern Spain. So, as we can see, the Daphnia are all over the place, and I actually see a road here, so this is near civilization, so you might not actually have to go into the middle of nowhere to find them. Okay. Under favorable conditions, they can propagate asexually for many years. In unstable environments, when environmental conditions deteriorate, Daphnia are able to switch to sexual reproduction. In this case, populations produce first males, asexually, and then haploid eggs that need fertilization. Production of sons and haploid eggs is regulated on the population level, with some genotypes never producing males. Fertilized eggs start development, but then undergo developmental arrest. Resting embryos require a dormant period before they continue to develop, resulting in only female hatchlings. The resting stage, also called epiphyum, refers to the entire resting structure, the shell and the resting embryos they contain. Because the embryo is still encased in an eggshell and has an egg shape, it's often called the resting egg, but this can be misleading. Some lines of Daphnia are known that are able to produce resting stages entirely asexually. So when conditions get rough, deteriorate, they might need to make some resting eggs sexually, so they can, I guess, have eggs that last until conditions are more favorable. Outside in says, anyone found Daphnia in the wild in chat? Not sure about in chat, but there is someone near me that brings stuff like Daphnia and fairy shrimp that he catches somewhere. I feel like he works in like water treatment or something. He's always bringing live foods that he collects, though. He did a talk on it, but I was late, so I missed most of it. What's up, Maven? How's it going? And what's up, Scotty? Jesse, I see Jesse popped in. So here we see the life cycle going on here. So there's a lot of arrows, so I'm going to do my best. I'm going to do my best to follow it. But here you can kind of see the twisted family tree of Daphnia. Steve said was late. Shut up, Steve. Captain says, I have found Daphnia while on a hike. Wow. Mark says, I've collected some eggs, dried them, and refrigerated them, but have not revived any. What, you saving them for a rainy day? Those locations were only a few hundred kilometers away from me. See, Mick, I knew they were in your area. Now, if only I knew how much a kilometer was. Jacob said, I put a 30-gallon drum outside in the summer, and Daphnia always appears, and I culture it all over the place. Oh, that's pretty awesome. So here, okay, I'm going to try and follow these arrows. So I think here we have what? Female. Female can produce a parthenogenic daughter, which then can continue the cycle. So this is like the asexual reproduction circle here. But then here we have some haploid egg formation, which seems to involve... Okay, we got a parthenogenic son. Okay, we got haploid egg formation. We got the males over here. We got mating. We got the resting eggs, the sexual eggs. They're in diapause. Then they hatch as a sexual daughter when conditions are favorable. All right. Steve said, showing up 20 minutes before it ends isn't late. You missed it. Holy. In the textbook description of the Daphnia life cycle, sexually produced offspring hatch from resting stages in the spring. So that's something I'm like keeping in my head is kind of interesting. Just that, okay, they do the sexual reproduction to make the resting eggs for when they need to, I guess, when conditions are bad. These offsprings then reproduce asexually across the season and finally terminate the planktonic phase by producing resting stages that endure the harsh winter conditions. However, studies in different habitats have revealed that this is the yearly life cycle in temperate water bodies common to Central Europe, where this model system was first developed. Depending on the species and habitat, any ecological aspect of this life cycle may differ. For example, Daphnia may continue in the planktonic form throughout the winter or may persist in short-lived desert rain pools or tiny rock pools. Populations may only be active in winter, parentheses raining season, produce no resting stages or produce resting stages asexually. The commonality among all Daphnia is that asexually reproducing animals are found in habitats where water temperatures are at least for part of the planktonic season between 10 to 30 degrees Celsius, and that they're able to outlive harsh conditions such as temporary dryness, freezing, and periods of low survival probability. You ever been through a low survival probability period in your life? Haven't we all? Who knew? Branchopodia were so relatable. In the form of resting stages can survive many years in pond sediment, and they tolerate drying and freezing. Mark says, as a matter of fact, I am saving them. If I get tired of caring for them, I could bring some eggs out and start again for free. How do you collect the Daphnia eggs? Outside is that felt that. Steve said I had a 10-year stretch of low survivability. See, come on, man. We could almost be Daphnia. Can y'all tolerate drying and freezing? We might be onto something. Daphnia can be collected from their natural habitats either as planktonic animals or in the form of their resting stages, which are often found in the sediments. Look, asked a question, answered. Planktonic females can be cloned naturally, enabling researchers to produce unlimited numbers of clonal offspring and keep genetic lines for many generations under laboratory conditions. They're kept in either natural freshwater or commercial mineral water or artificial medium and are fed mostly unicellular green algae. On a commercial scale, Daphnia are even farmed as fish food. Okay, now we're talking. 15 to 22 degrees is a good temperature for asexual propagation for most species. Resting stages collected from pond and lake sediments or isolated from sliced and dated sediment cores can be hatched by exposure to oxygen-rich water under daylight and ambient temperatures. Hatching success, however, often low, especially for old resting stages, for reasons we do not yet understand. Although genetic effects, deleterious mutations certainly play a role. Now, if you ever order from Greenwater Farms, that's probably how they're getting their Daphnia. There's a place with some sediment that's got the Daphnia resting stages. They're scooping it up. You know what I mean? They're putting it in, and maybe if you had a bad hatch, you just got unlucky. That was an older resting stage, perhaps. But I know a lot of people have done Greenwater Farms. For the most part, their stuff's worked for me. The worst one I had was I ordered some, what was it? It starts with a T. We just talked about them, the little crabby things. Well, the vials just had no eggs in them, so I can't say the hatch rate was bad, but there's just no eggs in the vials. Captain said the resting cysts create males and females to reshuffle genetics. Typically, males make cysts when conditions get bad. So reshuffle they can benefit or not when they hatch. Mark says collect them in the edges or in the detritus. Dry them out on a paper towel in the fridge. Okay. Mick called it Shirley Cheryl with an S. All right. Okay, buddy. Get some rest. The egg castings can be harvested with a net. If you harvest them in a bare bottom container, the longer you have the culture, the more eggs they'll drop. Okay. Ah, yes. Triops. Thank you. Outside in. And then, of course, Fathom reveals he's in the chat just to correct me like a true friend. All right. And Captain says can't confirm they do hatch out. Fantastic. So clonal reproduction allows us to blah, blah, blah. The scientists talk. Let's get to the things I want to hear about. Come on. Daphnia magna currently being the most used species for routine crosses. It's possible to cross females with males from the same clone, resulting in genetically selved offspring. This combination of sexual reproduction, outcrossing, and genetic selfing, which is mother with clonal sons, and clonal propagation allows researchers to conduct powerful genetic studies and create genetic panels that can be kept by clonal reproduction for many years. All right. Wow. They says that Daphnia research goes back several hundred years. They didn't have anything better to do, like surviving, I don't know, tuberculosis or something. Really? Daphnia got some attention. Okay. Okay. Wow. Uh, uh, I'm not even gonna try and say this name, but they studied macrophages in Daphnia, and they won the Nobel Prize in 1908. I don't know if it was for Daphnia, but the Nobel Prize winner was interested in Daphnia like 100 years ago. So, come on, we might be on the path to greatness here. Fathom semi covers bro. Back to my fake account. Yo, I think Fathom might be emoji man. I'm not gonna lie. He might be emoji man. One second, let me turn my light back on. Turn the lights in here, baby. Extra bright, I want y'all to see this. All right. Okay. So this was good. I think the most important thing here is gonna be about the environment and stuff like that. We're gonna get into the fish nutrition and specific stuff in the next few ones, but I just want to establish what the hell these things are. And also, one interesting thing with Daphnia that I kind of want to get into is what this part of it is, the host-parasite interaction, and some of the things they eat besides unicellular algae, by accident maybe. Because I've been really interested in parasites recently. I have. Green water rotifers did not hatch for me. Oh, man. I'm not dumb enough to be that guy. Hilarious. What's up, Marshall 2007? How's it going? So in their natural habitat, Daphnia are often infected at high prevalence with various parasites, including viruses, bacteria, microsporia, fungi, nematodes, cestodes, and others. While there's more. Many of these parasites can be co-cultured with their host, opening the door for experiments that examine host-parasite interactions on the individual and population levels. Compared to many other host-parasite systems, the Daphnia microparasite system offers a level of experimental control that's unsurpassed, enabling us to test basic models of parasite ecology and evolution such as mass action principle, evolution of virulence, host and parasite evolution and coevolution, the effects of aging and host-parasite interactions, infections with multiple parasites, and parasitism, and the face of predation and other stressors. Oh my gosh. And also, of course, they're transparent. So many infections can be diagnosed from the outside without killing the host. So let's see what some of these parasites look like. Oh, I'm already scared. So from A to D, we see some bacterium infections. So figure A, infected is right and the uninfected is left. All right. So the uninfected is left and the infected is right. Okay. They both look kind of gross up close. So let's see. Bacterium Pasteurella ramosa colonizes the body cavity of the host. So that's what this one is. I guess, I mean, looks pretty full of something gross there. So adult Magna will eat paramecium and young copepods. Okay. Outside in says parasite heights in small spaces, top 3 fears. They also eat paramecium. Okay, everyone is aware that they're eating paramecium. Okay, we're on the right track here. B is transmission stage of the spores of P. ramosa. And then C and D is the attachment of green fluorescent labeled P. ramosa spores to the oesophagus and the hindgut of the host. So this is all trying to track the spores of this bacterium. So you can see them infected in here. This is very up close and personal. Okay. So, what else do we want to get through in here? I think we might be reaching the end of what we need to look at. So, Daphnia. We got some resting stages. We got some reproductive information. Let's see what we can get into next here. I think I know. So this is going to be species specific, and it's going to also include something that many of you will revel in the fact that I will have to talk about since it's in this paper, and I do hate it greatly, but unfortunately they're being looked at together. for some of the same use, and that is Gamorous. I'm not even going to grace it with the mention of its common name. I'll refer to it as Gamorous. Okay, the Gamorous is in here. Daphnia magna and Gamorous pulex, novel promising agents for biomedical and agricultural applications. So we're getting a little bit closer to the fish stuff. All right, we're gonna make our way there, or we're gonna keep it a little bit more general till we get there. Mick said, "I will never kill my host." Well, rarely. Oh my gosh. Captain said those scientists did those Daphnia dirty. Yeah. Let me infect you. Let me make the bacteria that infects you glow green so we can see it inside of you better. All right. So, this tested Daphnia magna and their effect against pathogenic microorganisms and their effect on plant germination. All right. So they're looking at the antibacterial. Now I've added some Daphnian stuff to some egg trays and stuff with my aquatic isopods. Like, I've been kind of on this thing of like, what are the natural predators of some of the things that cause the fungus in eggs? So I've been kind of desperately doing these things. So I'm kind of hoping that there's actually something to what I've been doing with my delusions. Oh, what's up, Johnny Best, with the $5 donation? How's it going, Johnny Best? Thank you very much, my friend. With the holy. Yo, $5 can get you a lot of Daphnia. I'm just saying, if you know the right guy, Thank you. So zooplankton plays a critical role in aquaculture, influencing survival of livestock under harsh conditions. Zooplankton contributes to the aquatic food chain as an intermediator, feeding upon bacteria, fungi, and algae, while turning fed by numerous invertebrates, birds, and fishes. Zooplankton are used as environmental indicators, and recently plankton biotechnology has gained extensive significance because of its rich constituents involved in active primary and secondary metabolites. I'm going to look up this word, metabolites. Metabolites act as signaling molecules. Okay, serve as cofactors, energy production, and storage. Okay. The water flea Daphnia magna is a microcrustacean zooplankton widespread in freshwater bodies. Recently, it was used for fish fry in aquaculture as it has a high nutritional composition that varies according to the culture medium and viability degree of phytoplankton. This is one thing why reading the general stuff can be good for what you actually want to find, because now we have all these helpful references about using Daphnia to feed fish fry. So they reference the effect of feeding with different types of nutrients on intensive culture of water flea Daphnia magna, and that's something in 2020. What else we got? Ooh, anything else with the fish fry? Production of Daphnia similis claws using wastewater from tilapia cultivation in a biofloc system. Okay, that sounds a little bit interesting. Let me just Google this one real quick because now I'm curious. And this one doesn't talk about scuds. It's not because of that I'm trying to, check it out. Captain Critters, what is your website name? Fathom says organisms that metabolize. Thank you for that. Thank you. We got the science translator on deck. Okay, here we go. The effect of feeding with different types of nutrients on intensive culture of water flea Daphnia magna. No scuds in sight, but the scuds did help get us here. Okay. These experiments have been done, aimed to prove that water flea Daphnia magna can be intensively cultivated and grown on a variety of food industry residues, as well as algal powder under controlled culture conditions with no deficiencies. Let's see. I'm going straight to the materials and methods, and this is in Egypt. So Daphnia magna was isolated from Lake Mariot, one of the Egyptian northern lakes. A comparison was made between the results of the use of the different nutrients to determine the best among these in the terms of breeding. They were raised on spirogyra algal powder, rice bran powder, wheat bran powder, yellow corn powder, and soybean powder. And my tank says, "Are you selling Daphnia?" I am not. But Captain's Critters, she just posted her link. She's selling Daphnia. Yep. So she's got some coupon codes there, so feel free to use those. I don't know why she'd be giving discounts. Didn't even offer me a cut of them. You know what I mean? Just giving discounts, bro. Tax them. I'm joking. But big ups to Captain. Mick posted wiki in biochemistry. A metabolite is an intermediate or end product of metabolism. The term is used for small molecules. Metabolites have various functions, including fuel, structure. All right. Food particles have been micronized to 60 microns to be suitable for efficient filtration by Daphnia. Micronized operation was prepared and treated by hand mixer. Weighted quantities of feed powder were suspended in water and added manually 2 times a day. Oh my gosh, man. Precipitations were siphoned every 2 days to exclude them from the bottom of the tank to prevent anaerobic conditions. So they raised them in transparent tanks, 10 to 99 liters, 12-hour daylight, 12-hour fluorescent light tubes. Culture tanks received illumination of 5,000 lux at the side of these transparent tanks. Oxygenation of the tanks was assured by air bubbling. Aeration caused water agitation, which helps in the regular distribution of organisms overall. The culture water body was following the method of Ding's 1973. What type of creatures are seed shrimp? Are they copepods? Mix on Google, yeah. Captain said Daphnia being raised on tilapia poop water who were raised on sewage water is an interesting infinite food glitch. Well, listen, the thing with everyone with the live food stuff and fish, you want to try and close the loop as much as possible. And feed this duckweed, feed that thing. Like I'm feeding my mealworms duckweed. Maybe I'll feed my mealworms to my fish. Maybe my fish poop water does make good green water. And maybe I do feed my Daphnia off of that. Maybe I start eating Daphnia. The opportunities are endless if you have no boundaries. Jacob said, I think they're ostracods. Okay. Seed shrimp, maybe ostracods. Well, there'll be a seed shrimp alive eventually. Oxygenation, okay, yep. Replacement, 25% of tank water was performed on a weekly scale by using dechlorinated water. The environmental parameters were adjusted where the temperature was set as 22 degrees Celsius, salinity at 0.5%, ammonia 0.02, pH 7.8, and oxygen was 5.5 milligrams a liter. These parameters regularly measured twice a week to ensure it does not change. The cultivation tanks were left without harvesting for 5 weeks. Damn. To increase the cultivated numbers, and after that, the harvest was done on a weekly basis. The selectively harvest was performed by using 1 millimeter screening net to harvest the large size individuals. See? The neonates and smaller individuals were returned to the cultivation tanks. Calculation of feeding rate. I'm not going to be able to understand that. That's math. Chemical analysis. Ooh, this is what I'm looking for. Daphnia magna chemical composition. Moisture, crude, protein, crude fat, carbohydrate, calories, fibers, and ash was analyzed according to the standard methods of AOAC 1990. Moisture content was estimated by drying the samples to constant weight at 70 degrees Celsius in a drying oven for 48 hours, while nitrogen content was measured using an automatic Cadal system. And crude protein was estimated by multiplying nitrogen content. Lipid content was determined by ether extraction and a multi-unit extraction Sauclet apparatus. Ash was determined by combusting dry samples in a muffle furnace at 550 degrees Celsius for 3 hours. Jesus, is it cooked yet? I mean, my God. What is a muffle furnace? Wow. This looks less scary than I expected. The muffled furnace, I was thinking like, throw him into the fire. This is like the mighty toaster or something. I'm a little bit disappointed. I am. Okay. Determination physiochemical characters of the culture medium. Water temperature was measured by using a mercury thermometer. All right. Turbidity of the water was measured by a turbidimeter bench. Hydrogen iron concentration pH values of the water were measured by using a digital pH meter. The Levibon Senso Direct pH 200. Ooh, you got the Nimbus 2000. This is it. This is it. This is $580. No way. Used. And it looks like this. They couldn't like put some, I don't know, some Louis Vuitton logos on it or something. So this science stuff needs some better brand. I just want to feel cooler, you know? What's with all the beige? I need like the black edition or something. All right. The dissolved oxygen was measured by DO-meter, the HQ30D fleximeter, HACH. Okay. Yeah, we've looked at this thing before. We've looked at this thing. It looks like a precursor to a Palm Pilot or something. This is bad. You guys remember Palm Pilots? Mick, you're old. Yeah. At least Palm Pilots could play like, I don't know, tank, Pong. There was like a racing game on the Palm Pilot. Man, Palm Pilot was lit, man. I'm not gonna lie. I had a Palm Pilot I used to play on. Had some fun games on there. Mark said, noticed morning are very sensitive to dissolved metals. Found using tap water conditioner very helpful. The bacterial content of harvested Daphnia was analyzed to ensure it was free of pathogens and its suitability for use in feeding fish larvae. The total count of bacteria and yields was calculated, and their contents of Salmonella, Clostridium, Listeria, and Escherichia coli were analyzed, as well as their yeast and molds content. Here's the vitamins and toxins content. All right, so they're looking at everything. So I expect a full conclusion on how good that Daphnia are for feeding my fish at the end of this. Vic said, perhaps it's just me, but I can't see Versace starting to sell those. Come on, Mick. Use your imagination just for once. All you gotta do is believe. You know? After 5 weeks of Daphnia magna cultivation, the maximum attained density was 10,000 individuals a liter from tanks feed with rice bran powder, followed by an average of 9,000 individuals a liter from the feed on soybean powder. It was noticed the average number of Daphnia population that feeds on algal powder was similar to the feeds on yellow corn powder, 8,000 individuals a liter. On the other hand, the population which was fed on wheat bran yielded the minimum average of population density among all experiment tanks, 7,000 individuals a liter. Hence, the nutrients used in the current experiment can be arranged in the terms of best in producing Daphnia to rice bran, soybean, algal powder with equal to yellow corn, and finally wheat bran. At week 1, there was no significant differences between the densities of the population treated with algal powder, wheat bran, yellow corn, and soybean powder, while all these treatments differed significantly with that treated with rice bran. On the other hand, the case changed at the end of the final week, as there was no significant differences between the density of populations resulting from the nutrition with rice bran and those fed on soybean, which had been differed from the rest of the treatments. So here we see the numbers throughout the weeks. Captain said they're literally feeding it poo-poo bacteria. Fantastic. So week 1, we see that they are kind of similar. Soybeans at the top, but they're all within 1,000 or so of each other. Week 2, soybeans still ahead, but we see rice bran in second with 4,000 individuals. Week 3, we see a big jump in the algal powder. It doubles from week 2 to week 3. Rice bran at 7,000, soybean at the top with 8,000. Okay, the week 4, things are starting to pick up a bit. Rice bran 8,000, algal powder 7,000, soybean at 8,000. So there's a tie between rice bran and soybean powder at week 4. But then in week 5, rice bran powder comes to the top at 10,000, with soybean powder at 9,000. Yellow corn tied at algal powder with 8,000. And wheat bran powder with 7,000. To be honest, a lot of these are very similar. You know what I mean? Like some of the differences didn't appear until the end. So we'll have to look at the nutritional composition, because for my own purposes, it's not like, I'm not going to use the one that was the lowest, but all of them seem to produce a decent amount of Daphnia. So like, what's the problem? You know what I'm saying? Okay. So the nutritional content of Daphnia that was isolated was analyzed to determine its efficacy as a food for fish larvae, as well as the difference between the concentrations of its contents from those used in other studies. The obtained data showed that the protein content of Daphnia magna was 47.7% and fats was 6.1%, while ash and carbohydrate recorded 15.6% and 20% respectively. On the other hand, calories recorded 391. Okay. Chemical composition, nutritional values of Daphnia magna. Percentage of its dry weight. All right. So yeah, here we have the chemical composition. Ash is 15. We got the protein 47, just like it said. We have the feeding rate here. In the present scale experiments, culture water volume 10 to 130 liters, Daphnia population densities can rise up to 10,000 and more individuals per liters within 5 weeks. The average amount of weekly harvest after the 5 weeks was 450 grams wet weight a week. The species has been intensified, and the product was harvested and dried at 50 degrees Celsius for 24 hours. The obtained dry weight does not include the weight of small-sized individuals lesser than 1 millimeter or the eggs, because they used the 1 millimeter net, so anything smaller they wouldn't have even been catching to dry off. All right. The cultivated individuals reach maturity after 7 days at a length of approximately 1.5 millimeters and harvested 1 millimeter. Okay, yep, on a weekly scale. This table's making me hungry. Really, Mick? Really? After harvesting the cultivated adult individuals, the average number of the remaining small individuals, which remained for starting the next week, was 600 individuals a liter. The calculated total amount of food that gives daily was 0.18 G/L/D, which is the equivalent average of 180 G/M3 a week. The obtained average Daphnia wet weight harvest is 450 grams a week. The average corresponding conversion ratio from food to Daphnia was 2.4 to 1. This means that 2.4 kilograms of food powder can produce 1 kilogram of wet Daphnia biomass. The obtained yields were between 400 and 490 grams wet weight a week, with corresponding food conversion ratios between 2.55 to 2.27. It was noticed that rice bran powder gave the highest productivity as well as the highest food conversion ratio among all foods used. Okay. And they didn't really find anything bad as far as bacteria in them, it looks like, except for listeria. Let's look at vitamin content. The obtained results show that the farmed Daphnia magna contained most of the vitamins necessary for the growth and improvement of the health status of fish larvae. They contain vitamins B2, B6, B12, A, D, E, and folic acid in good proportions. It was also surprising that these organisms contain tannic acid and beta-carotene, which are considered the most important antioxidants. It was also observed that these organisms contain very low levels of the toxic compound aflatoxin. And here you see the values of them all there. That's making you hungry. All right. So, although harvesting large sizes... One second. Although harvesting large sizes is a stressful and time-consuming process, it carries many benefits as it gives an opportunity to produce young as well as increases production by more than 45%. Harvesting for large sizes and leaving small ones leads to many benefits. Ooh, okay, hold up. New strat alert. Such as improving the characteristics of the culture environment and stimulating the remaining species to reproduce, which leads to an increase in the numbers of ovigerous females. This phenomenon was observed both by Hezig in 1979 and D. Paul in 1981, who noted that leaving the farms without harvest for a long time leads to reducing the numbers of offspring due to the increase in the length of time needed to reach the reproductive stage. In general, the yields attained using the current food items are somewhat promising. Okay. They're higher than results obtained by Evleva in 1973, who used horse manure and obtained a result of 350 grams a week, as well as DePaul in 1980, which used pig manure, which only did 35 to 250 grams a week. Others who used microalgae in nutrition produced various results ranging from 100 to 700 grams per week. Okay, interesting. The aflatoxin B1 values within these organisms were very low. It's less than the permitted values of the United States Food and Drug Administration. Okay, these organisms contain high levels of important vitamins as well as two of the most important antioxidants, tannic acid and beta-carotene. In addition to these organisms are considered free from many pathogenic bacteria, which makes the use of these organisms as food for fish beneficial and safe. All right. So, one data point to add to the wall for the fish farming of Daphnia magna. JP Jeffrey said I'd go bat guano. Really, JP? Really? All right, let's get back. So, I did get a little distracted there, but like, I like to. This is how I read things. So we're originally reading this Daphnia and Gammarus thing, but now I saw the reference. I want to go check out the reference. So we went and checked out the reference. Okay, it is what it is. This is how we go. But if we go back to where we were, if you can pick in your brain, Daphnia, Magna, and Gammarus pollex for biomedical and agricultural applications. And what's up, Beto? How's it going? So the water flea, Daphnia magna, we're familiar with at this point, what it's about. But they're pairing it with Gammarus here. Gammarus, they say, has a central role in organic matter degradation. You could say that again. And risk assessment. Besides its contribution to the food net, it contains high levels of protein, fat, and amino acids. Thus, Gammarus meal enhances the feed intake, immune response, stress resistance, and growth performance in fish. Gammarus is used as a cheap alternative substrate for animal protein in the diet of highest value fish. Therefore, it's important to explore the effect of Daphnia magna and Gammarus pollex biomass against the most common pathogenic microbes that could infect either the fed fish or humans as the end consumer. So this might be a higher standard than even for feeding our fish, because this is talking about feeding them to things that will be fed to humans eventually. All right. It's important to study their effect on plant germination because the drainage water of fish farms is loaded with biomass and is recycled for irrigating plants. Pathogenic microbes and infectious diseases are one of the major problems in the medical field, leading to the death of many people worldwide. Antibiotic resistance is counted as one of the most parameters affecting human health, particularly after the emergence of multi-drug resistant pathogens. The wrong use of antibiotics and the deficiency of capabilities and scientific tools to improve such drugs has led scientists to explore alternative natural molecules with potential antimicrobial activity. Several types of bacteria and fungi can enter individually or together, a vital matrix called biofilm, through a microbial molecular communication system that regulates the virulence traits of these microbes and introducing infections by biofilm-forming microbes via medical devices. The biofilm is a complex network of lipids and proteins used to protect microbes against a range of antibiotics in the host immune system. Thus, it's necessary to search for new treatment substrates, such as natural molecules, to provide a new vision in treatment and limit the spread of multi-drug resistant pathogens. Different species of fungi cause major plant diseases, mostly controlled by synthetic fungicides, whereas repeated fungicide leads to rapid evolution of fungal resistance against fungicides, creating a high demand for exploring a natural alternative. Come on, man. Come on. Let's get the bat guano rolling. Okay. The discovery of natural antimicrobials might meet the consumer demand for environmentally friendly materials, avoiding chemicals with harmful side effects. There's been a promising trend for scientific research in the industrial applications of biotechnology and marine compounds. So in this study, for the first time, we looked at the antibacterial and antifungal and antibiofilm activities of the dried biomasses of G. pollex and G. magna to be used safely in the fish diet and medical field and limit the spread of all the disgusting things I just listed. So here we go, materials and methods. My goodness. So they collected Daphnia and Gammarus from the northern Egyptian Lake Marriott. Mixed in, now we're talking true science lesson. The Cladoceran species Daphnia magna and the amphipods Gammarus sp were collected with a standard plankton net of 200 micrometer mesh size, which was lowered vertically to a shallow bottom, then pulled up to the water surface. Then the net content was placed into plastic containers filled with lake water and identified the species as Daphnia magna and Pulex to a dissecting microscope. The samples were then identified by traditional morphological and biochemical methods. They kept them in 1,001 transparent plastic holding containers that had dechlorinated tap water with a salinity of 8% as in nature for 24-hour acclimatization before starting the cultivation experiments. After cultivation for a certain period under standard conditions, according to the methods described by Abu Talib et al., both species were dried at 70 degrees Celsius for 48 hours to be ready for further experiments. So, antibacterial determination. The antibacterial activities of Daphnia magna and Gammarus pulex were evaluated using an agar well diffusion assay on Muller-Hinton agar plates, according to the CLSI guidelines against human pathogenic bacterial strains. Pseudomonas aeruginosa and E. coli, there we go, which represented gram-negative bacteria, whereas Bacillus subtilis and Staphylococcus aureus were used to represent gram-positive bacteria. The inhibition zone diameter was specified in millimeter. The micro-dilution method in microteter plates was applied to determine the minimum inhibitory concentration of active Daphnia magna and G. pulex against the pathogenic bacteria. All right. Overnight cultures of the test strains were added to the broth media dispersed in the wells with and without Daphnia and Gammarus. Tank Book said, is he casting spells? Yeah, man. Yeah. Leviosa. Crucio. Giticus, Ophicus, Mystremicus. That'll teach him. All right. They performed all experiments in triplicates, as I was saying. Anti-biofilm assay, ASSAA. I gotta look this up because...
Assay.
Really? Assay? This is a terrible word. The testing of a metal or ore to determine its ingredients and quality, to determine the content of quality. Archaic attempt. I essayed a little joke of mine on him. That sounds... Yo, what kind of vocabulary are these people using? Black magic? Yeah, that's right. I'm gonna have to get Auntie K up here. She'll teach us some actual spells. You know? Okay. So they tested them against a bunch of biofilm producing strains as well. They were loaded into a flat bottom MTP containing tryptic soy broth media, supplemented with 1% glucose and mixed well. The tested strains were cultured and incubated overnight in TSB at 37 degrees Celsius, and after incubation diluted to reach the turbidity of 1.5 times 10^8 CFU. Why am I reading this, guys? I don't know what this means. Then inoculated MTP wells containing different concentrations of tested biomasses and incubated at 37 degrees Celsius for 48 hours. All right. Let's see what else. So the antifungal activity. Yep, they tested this by putting the powder of the Daphnia and Gammarus separately into wells of plates, incubating them for 5 days at 30 degrees Celsius. Antifungal activity was evaluated by measuring the diameter on the inhibition zone for each strain. Each strain was tested in triplicates. This is like a lot. This is like a high effort study going on right now. They're doing a lot. This is just all the different tests that they're running. We also have the seed germination test. Okay, I need to get to the results because I can't even comprehend what they're talking about right now. Beto says, I'm dabbling in biofilm, was doing for my loaches. Oh, very nice, very nice. Mikim said, microbiology science is in deep is a lot to learn. We just want to know the antimicrobial and biomedical and agricultural applications of Daphnia magna and Gammarus pulex. Like, can we keep it simple here? You know? So the chemical content of the two tested animals was analyzed to determine their prospective efficiency for industrial applications. Biochemical composition of Daphnia magna and Gammarus pullix is given in Table 1 based on 3 independent readings. The results point to higher chemical content of Daphnia magna in protein, fibers, nitrogen-free extract, and both types of energy, while pullix had a higher chemical content in fats, ash, and carbohydrate. Yeah, scuds are trash, bro. That's all you needed to know from that. Based on a high content of protein and other ingredients, Daphnia magna and pullix are proper alternatives for fish meal and feeding fish larvae. As far as vitamins and antioxidant content, they both contain vitamins and antioxidants required to improve the health status of fish larvae. The vitamins contained, we have a nice little table here. We have Daphnia, we got vitamin A, B2, B6, D, E, folic acid, beta carotene, and tannic acid before, whereas the Gammarus pullix had vitamin A, B2, B6, B12, D, E, folic acid, and beta carotene and tannic acid as well. Hardest working, let's go. The vitamin content in Daphnia magna attained from the study was lower than that mentioned in a previous study, although the antioxidant contents were higher. On the other hand, the vitamin and antioxidant contents of G. pullix of the study showed a lower variation than mentioned by a previous study. So, I mean, that could be diet or something. You never know. The results indicated that the Gammarus pullix had better inhibitory effects against gram-positive and gram-negative pathogenic bacteria than D. magna. The inhibitory action for D. magna and G. pullix biomass may be due to the reaction of biomasses with bacterial protein, leading to the inactivation of proteins and bacterial growth. Okay, so they're testing them against each bacterial strain. Results showed that the MIC for D. magna was 2.0 milligrams per milliliter against both P. aeruginosa and S. aureus, whereas G. pullix was also the same. Hence, biomasses had the same activity against gram-positive and negative bacteria. However, the mechanism of the bacterial growth reduction still needs further studies. Okay. Jesse said, sound or just me? My sound should be working. Smart person says, makes sense. Scuds with more fat-soluble vitamins if they have higher content on the whole. Yeah, stop muting your phone. All right, anti-biofilm activity. Gamburin 419 says, morning all, good morning where you are. It's currently 10:38 p.m. here, but you know what? It feels like a good morning, doesn't it? In this work, the anti-biofilm activity of the tested biomasses exhibited varied effects against different bacterial strains. Okay. G. pullix and Daphnia magna biomass affected the biofilm formation of Pseudomonas aeruginosa at concentrations lower than the inhibitory concentration. While the biomass didn't exhibit any anti-biofilm activity against S. aureus, MIC values of the D. magna and G. pullix against P. aeruginosa and S. aureus were 2.0 mg. Okay. The sublethal concentrations of D. magna and G. pullix exhibited anti-biofilm activity against P. aeruginosa with significant differences in dose-dependent matter where the concentrations of Daphnia magna were reducing the biofilm. Okay. 3:30 in the UK. 03:30. My volume is a bit low, Sage says. All right. I mean, I can increase the volume. I can turn things up if you wish. If you would wish, I can. I also have a limiter on, so let me make sure I didn't. Yeah. Let me know if that's better. Ole. The conclusion, though, I'm going to just read the conclusion at this point because I think I've tested my brain enough as it is. But in this study, isolated biomass of D. magna and G. pullix showed obvious ability to inhibit several pathogens involving gram-positive and gram-negative bacteria, as well as inhibition against some plant pathogenic fungi. The current report considers the first biotechnological study of the current micro and macroinvertebrates. The biomass from D. magna and G. pullix exhibited the possibility of applying them in the medical and agricultural fields, limiting the spread of some pathogenic microbes and enhancing plant germination. Fascinating. And that's just drying them up and grinding them up. Like, what in the world? 4:30 a.m. here in Sweden. Mick said, night owls are a thing. Don't I know it? Don't I absolutely know it? Okay. Well, where should we go next with this whole Daphnia adventure? What do you think? What do you think? Let me see. Let me look at my list here. So we could look at Daphnia magna as biological harvesters for green microalgae grown on recirculated aquaculture system effluents. Design and optimization of a recirculating aquaculture system for live production aquaculture, a case study using Daphnia magna. And this is sounding fascinating. Oh, yes. This is sounding quite fascinating. Let me see. I don't even know what to choose. Recirculating culture using Daphnia sounds good. What a bunch of nerds. I knew you guys would want. I knew someone would pick that one immediately. I absolutely knew it. Let me see if I can get the PDF for this real quick. Propagation and beneficent hobbyist / fish breeding. You're asking too much. JP Jeffries says it's all too good. Yeah. Don't I know it? Don't I absolutely know it? I also feel bad because like we missed new arrivals last week. I didn't do any of the auction review stuff. Like, man, I wasn't able to. This weekend is going to be hard for me. The next few days will be hard for me to stream as well. So I was like, I have to get a stream out right now. All right, just give me 1 second. I'm just trying to figure out what I actually feel like reading and what page wants to actually load for me, you know? Next thing, what's up, this smart person? Well, you guys aren't interested in Daphnia magna as an emerging environmental model of neuro and cardiotoxicity of illicit drugs? Okay, I'm going to have to subvert expectations here, but I don't think you'll be mad at my selection. How Daphnia magna defends itself against predators. Mechanisms and adaptations in a freshwater microcosm. Come on! Come on! Just finished refreshing my morning culture, so I figured to see what Nick was up to. You already know, man. Getting some devious Daphnia going on here. The devious Daphnia. Well, let's see how the Daphnia can defend itself. That's the question. The freshwater flea Daphnia magna is a prey for numerous predators. Ooh, numerous predators. Yet it possesses a wide range of strategies to defend itself against predation. Captain said, my bet is their defenses are round and hard to fit in the mouth. Okay, we'll see how that pans out. The aim of this work is to investigate the defensive mechanisms employed by Daphnia magna to reduce predation by the colinterate Hydra viridissima and two planarians, Polycelus felina and Dugicia gonocephala. To do this, we used a freshwater microcosm. An additional aim is to investigate interactions with the presence of the isolated endosymbiotic algae from green hydra, thus combining and observing this interaction of the zooplankton and microalgal component. Each experiment includes 5 replicates, 13.5 degrees Celsius and 25 degrees Celsius in crystallizing glass containers, 60 milliliter volume, 60 millimeter diameter, 30 millimeter height, including satiated, fed with larvae of Artemia solenia and starved predators. So some that have eaten and some that hadn't, respectively. 1 or 5 individuals of a particular predator species in a microcosm, as the isolated microalgae are unique, We tracked the 3 following mechanisms of Daphnia defense for the first time, including precisely this microalgal component. 1. Grouping, visual magnification, i.e. 2 or more Daphnia holding together. 2. The phenomenon of overproduction, any number of Daphnia in one container above the 10 initially added individuals. And 3. Accelerated movement, bullet movement, high-speed movements, in particular microcosms. This result provided new information for a better understanding of the interspecific relationships and systems that include both zooplankton and microalgal components. Gonocephala, that's a great insult. What? Daphnia defense group. What kind of microorganism naturally are the first to start in all aquarium setups? Ooh, that's a good question. Probably some kind of bacteria or something, right? Captain's Critter said, Daphnia, why do I hear boss music? That's hilarious. That is hilarious. So they used the green hydra and the planarians. Some planarias. Okay, stinging tentacles. Yep, we're familiar. Let's see the materials and methods. So we know the organisms. We know kind of what they've got going on. We know the containers they're using. In each microcosm setup, either 1 or 5 individuals of a particular hydroplanarian species were added with a dropper. In microcosms with more than 1 hydra/planarian species, hydras and planarian... I need some water. Oh my gosh. Were added ratios of 1-to-1 or 5-to-5 individuals. Whew! Gambirian 49 said, never changed your channel before, but YouTube knew the subject matter was interesting to me. Now I have another subscriber. I'm keen to see if you made it a fish room tour. Oh, welcome. Well, hopefully it continues to be interesting. You guys get too bored by the random stuff we read related to the topic. But as far as fish room tour, I haven't done a fish room tour, but I live stream in the fish room, and I often do show stuff around. But our room is kind of a mess, so not quite ready for any kind of tour. Mick said, cephala, in biological terms, cephala refers to the head or head-like structure in organisms. Oh, gonocephala. I see. Okay. So, 1-to-1 or 5-to-5 individuals ratio. So 1 or 5 predators, individuals of a particular predator species per 1 microcosm, respectively. 10 individuals of the prey, Daphnia magna, were added per microcosm in the case of the experimental setups that included the addition of the microalgae, 1/4 of the algae content from 1 test tube, and 10 milliliters of aerated water was added with a metal spatula and homogenized by stirring with the spatula without changing the direction of mixing. After mixing, the suspension of algae was added to a 60 milliliter container previously filled with 40 milliliters of aerated water, and then the constituents of the microcosms were put into these containers. Changes in the Daphnia were registered by visual observation. The grouping of Daphnia magna considered any holding together of 2 or more living individuals, 2 or more individuals, some of which were alive and some dead, and 2 or more dead individuals that had once been alive during the experiment. Overproduction of D. magna was considered any number of D. magna in one container above the 10 initially added individuals during the experiment. Alive and dead individuals were counted. Accelerated movement or bullet movement describes high-speed movements. Okay, we understand all this. So this is one of the microcosms here. Oh my gosh. The poor Daphnia. So with the Daphnia, and we have the P. felina, so planaria. And here's the grouped individuals of Daphnia. So it looks like 3 Daphnia here, 2 Daphnia up here, and you got the 2 planaria in the middle. Planaria are a prey of Daphnia. Did I not have this realization out loud? Because that's not something that I thought about. Smart person said, maybe I'm reading the gono as a prefix for angle, so not what I thought initially. Because Mick was saying gonocephala. Does it mean D-head? I don't know. Gonopodium? I see what you're saying. Okay. Oh my gosh, look at all this data. Oh my gosh. I can't read that. Give me the words. So in the control setup with Daphnia magna, so I think that's just Daphnia magna with no predators, And everyone's saying welcome to Gambirian. Welcome to the nerdy fam of Nick's in My Tanks channel. Let's go. So in the control, no grouping, overproduction, or accelerated movement was noticed. In the control setup with Daphnia magna and the microalgae, overproduction of Daphnia magna was observed only after exposure of 24 hours at 13.5 degrees Celsius and 25 degrees Celsius. Accelerated movement was present only after 24 hours at the higher temperature, so it's 25 degrees Celsius. The setup with Daphnia, with added green hydra as a starved predator, showed a grouping of D. magna after 1 hour with 5 hydras at 25 degrees Celsius, as well as after 24 hours with 1 hydra at 13.5 degrees Celsius, and with 1 hydra and 5 hydras at 25 degrees Celsius. So all those conditions, they saw groupings of Daphnia magna. In the setup with Daphnia magna and the planaria as a satiated predator, a Daphnia grouping was present after 1 hour with 1 planarian at the lower temperature, and with 5 planarians at both temperatures. After 24 hours, the grouping of D. magna was observed with 5 planarians at both temperatures, and with 1 P. felina at 25 degrees Celsius. When the planarian was introduced starved in the experiment, the grouping of D. magna was observed after 1 hour and 24 hours with 1 planarian and 5 planarians at 13.5 degrees Celsius and 25. Man, the Daphnia are scared of the planaria. And this is kind of crazy because they said, and so when there was no predators, they didn't do this grouping and whatnot. They're really responding to these damn worms. The Daphnia can swim. I don't. They got them in a small ass container. I get that, but like, I'm just trying to think in my head how the planaria are getting the Daphnia. I feel like the Daphnia can kind of move better, but man. In the setup with D. magna and green hydra and the planaria satiated predators, so they fed them before, the grouping of D. magna was observed after 1 hour with 1 hydra and 1 planaria at both temperatures. I mean, how would the Daphnia know that the planaria had just eaten? You know what I mean? Planarians are fast when they need to be. Interesting. And Gambrians, thank you for the welcomes. Welcome. JP says Nick is an aspiring college professor and he thinks we're his students. Bro, I look at this as me trying to learn usually basic sciencey words. And hopefully either you guys are helping me figure out what the hell's going on or we all learn together. Definitely not teaching anything. Smart person said they have a venom if they get to grab the prey. That's a good point. I mean, Daphnia asleep, right? You know? So, after 24 hours, the grouping of Daphnia magna was present with 1 hydra and 1 planarian, and with 5 hydras and 5 planarians at both temperatures, with the presence of starved predators. So it just seems starved or not starved. There seem to be grouping kind of similarly from what I can tell. Overproduction of Daphnia magna when the predators were satiated was observed after 1 hour with 5 hydras. With the algae in the control, I think they said it was after 24 hours. Yeah. With both temperatures. But in 1 hour. Let's see. Okay. 1 hour with 5 hydras and 5 planarians at both temperatures. In the case of 24-hour exposure, overproduction was observed with 1 planaria and 1 planarian at 25 degrees Celsius, and with 5 hydras and 5 planarians at both temperatures. When predators were introduced into the experiment starved, overproduction of Daphnia magna was observed after 1 hour with 5 hydras and 5 planarians at the higher temperature. In the case of 24-hour exposure, overproduction of Daphnia magna was observed with 1 hydra and 1 planarian, and with 5 hydras and 5 planarians at both temperatures. In this setup, D. magna and green hydra and D. gonocephala as satiated predators. After 1 hour, the grouping of Daphnia magna was present with 1 hydra and 1 of the D. gonocephala and 5 hydras and 5 D. gonocephala. So, okay, we're seeing a lot of responses. I'm not going to read every single one because they did a lot of different ones here. We're going to get to the discussion. Okay, they got to figure where the overall statistically significant difference in the grouping of Daphnia magna is shown. No grouping of Daphnia was observed in our experimental setup with D. magna and satiated hydras. Okay. And in the case of starved hydras, the occurrence of D. magna grouping was lower than in the case of starved planarians. The influence of the two species of predators on the occurrence of Daphnia grouping in relation to the influence of one species of predator was greater and was observed to a greater extent when only one predator individual was present in the microcosm. The incidence of Daphnia magna grouping in the presence of the microalgae was lower than in the setup without the microalgae, showing their influence on the stability of the ecosystem. When one individual of a predator was present in the system, although the occurrence of overproduction was higher in the case of starved predators, overall, a statistically significant difference in the overproduction of Daphnia magna in the experiment is shown. Okay. I see the light on my green water tank went off, but this isopod keeps coming in the corner still. I feel like I've seen the same isopod many times. Excuse me. Okay, let's look at the discussion. At the level of aquatic ecosystems, freshwater zooplankton is a very common prey for other larger invertebrates. Daphnia demonstrate a wide range of defensive strategies against both tactile and visual predators. In the presence of predators that prefer large water fleas, a higher rate of reproduction, whereby water fleas produce smaller-sized individuals to avoid predatory attacks, is observed. The reverse situation is seen in the presence of predators that prefer the individuals of smaller size. The swimming speed of Daphnia magna in the presence of cyanobacteria toxin microcystin increases significantly, which can be used as an indicator to detect lower concentrations of this toxin. Okay, so kind of go into some other stuff that's known. So okay, let me go down a little bit. I want to go back to the figures. Yeah. I can't even. I'll be honest. It's hard for me to, it's getting to that level of hour when I see a graph, my brain says, turn it off. Turn it off. All right. Until recently, biological drivers of plankton aggregation were underappreciated. Our results show that the large grouping of Daphnia magna individuals was present as a defense mechanism against predatory attacks. Although in the setup with Daphnia and the different planarians, the grouping of D. magna was recorded in both the case of satiated and starved planarians. This was seen in much larger statistically significant extent in the case of the starved predators. They had a much greater tendency to catch their prey. Oh, God. In this case, the frequency of attacks of the P. felina and D. gonocephala increased. Such a grouping of prey individuals was very likely one of the defense strategies against predatory attacks in which D. magna camouflage themselves among dead individuals in order to avoid death, or simply by grouping to achieve a larger size, with which D. magna might attempt to intimidate the predator and make it abandon the attack. Similarly, the grouping of D. magna is visible in the middle of an experimental dish in the presence of a large number of flatworms, of the flatworm mesostoma. The grouping of a prey individual as a strategy for thwarting predator attacks has been observed in other species, etc., etc. In the case of starved planarians after 24 hours, there was a lower incidence of grouping of D. magna compared to after 1 hour because there was a smaller number of living individuals. What the hell, bro? I'm glad they explained why, but yeah, there was a lower case of grouping after 1 hour because too many of them were dead. Holy. But yeah, there's a lower incidence of grouping of Daphnia magna compared to after 1 hour, after 24 hours compared to after 1 hour because there was a smaller number of living individuals. Captain says they're throwing the younglings at the predators. You only have to be faster than the slowest among you, including your own baby. Well, I mean, the babies are clones, so it's kind of like throwing themselves at it, right? Like a smaller version of themselves. Predators like big bites, then you give up your elders. If the predators like big bites, you give up your elders. Okay. They formed a bait ball like krill. Yeah, don't people show off their Daphnia balls? Pause. Okay. A similar situation was observed in experimental setups with one individual of two species of predators was present in the system. In the setup with Daphnia magna and green hydra, the grouping of D. magna individuals was recorded to a smaller extent and only in the case of starved animals. A reason for this is that green hydra moved to a lesser extent and at a lower speed than planaria. Therefore, D. magna could more easily avoid getting caught by them by retreating to the parts that they weren't at in the dish. The difference was reduced when there were 5 predator individuals. When there was a large number of predator individuals, D. magna grouped to a lesser extent due to more frequent predator attacks. A lower incidence of grouping was observed when the microalgae was present in the system along with the predators in comparison to other microalgae-free setups. It's possible that grouping is hindered by the aggregation of microalgae and the formation of microalgal net, with water fleas trapped in a microalgal hunting net. Excuse me. In conclusion, with the presence of predators in this microcosm, individuals of Daphnia magna defended themselves by grouping in such a way that 2 or more living or dead individuals were grouped together. The functional diversity of Daphnia was also demonstrated in the overproduction mechanism. The regularity of overproduction was observed, especially in setups with Daphnia, polystylis, and algae, and Daphnia and algae. After 1 hour, there was no or low overproduction when predators were introduced into the system, either satiated or starved at both temperatures. Overproduction was observed to a greater extent after 24 hours with satiated and starved predators at both temperatures. This is probably because 1 hour of contact between Daphnia magna and other constituents of the microcosm was too short for possible overproduction. A significantly higher incidence of overproduction was observed when microalgae was present. Therefore, the microalgae had a great influence on overproduction in addition to predators. An example of functional diversity is also displayed by Daphnia pollux. If it's exposed to chemical stimuli that signal the presence of a predatory larva of the Deuteron triborus, embryos of Daphnia pollux were developed into individuals with defensive spines that would reduce their risk of being eaten by a predator. Captain was right. Captain said the spines on Daphnia pollux were why there were no fur, right? Didn't you say that? That's crazy. Can you imagine, like, you get a chemical, you smell your predator and you just develop babies that got, like, I don't know, an AK-47 built into them or something, like. Wait, microalgal hunting nets, the algae is hunting stuff too? Net. Microalgae form aggregations and nets to protect themselves from predation and attacks. Okay, we're gonna have to look at these microalgae nets. It's possible the group is hindered by the aggregation of microalgae and the formation of the microalgal net, with water fleas trapped in the microalgal hunting net. Yeah. Okay, this is a whole can of worms. Formation of microalgal hunting nets in freshwater microcosm food web microscopic evidence. I'm just going to very quickly, I'm literally going to go to the very end and just see. During the experiment, we observed the formation of microalgal nets. It's possible that microalgae, after aggregation, formed the microalgal nets in order to protect themselves. Given the control replicates where only a suspension of isolated endosymbiotic microalgae was present, both microalgal aggregations and microalgal net formation occurred. Okay. Occurrence of the net was observed in experiments most intensely when P. felina was present. The nets were macroscopically visible as green intertwined filaments. The nets were present in the microcosm after 1 hour for microalgae and microcosms with H, so they tested it with a few different predators. So yeah, microalgae makes these little nets, I guess. And it might slow them down. Captain says, part of me wonders they're just popping out babies based on temp and food availability. I mean, I think no, because they tested it. They did a control without the predators, and there's more overproduction with the predators. I think is how they ruled that out. Smart person said this paper has to be the same people as the Daphnia paper. I was about to say I agree, and it was on the same site. It looked very similar, so it looks like they had a couple things going on here. Yes, I was talking about the spines. Crazy. Whew. In our research, there was a greater incidence of overproduction when predators were starved. Then individuals of D. magna were under greater pressure from predatory attacks and resorted to overproduction. It was observed that in some cases, Daphnia magna individuals were smaller than the initial individuals added in the experiment. In the presence of predators that prefer larger individuals to Daphnia magna, there is a higher rate of reproduction, whereby Daphnia magna produces individuals of smaller size to avoid predatory attacks. The presence of predators can lead to a reduction in the body size of the prey, which serves as a defense against visual predators. Certain invertebrate predators have strict restrictions regarding the shape and size of their prey. Interesting. As for the concept of overproduction in research, it's not entirely clear whether Daphnia magna really multiplied, some of them being sucked out by planarians during predation, or whether these sucked out individuals of Daphnia magna were actually molts of individuals added. If it was a case of multiplication, Daphnia magna was capable of fast reproduction within 24 hours under the given predatory pressure. It's possible that the overproduction of Daphnia is a consequence of possible feeding of microalgae, since the period between moltings depend on environmental factors and may vary in adult individuals. In any case, clear that individuals of Daphnia magna under predation pressure reacted rapidly, either by reproduction or by molting. Okay, so whichever one it is, it's still the result. It still resulted in reproduction, seemingly. But with two different predator species, overproduction of Daphnia was possibly stimulated by the fear of predation by a larger number of present predators. Finally, if it was the case of molting, this would serve as a camouflage mechanism for the water fleas that survived. The accelerated movement, okay, yeah, we understand how that could be helpful. They observed this kind of movement when there's no predators, but within the presence of the microalgae system, in the setup of Daphnia magna with the microalgae, the greatest accelerated movement was observed after 24 hours at the higher temperature. Basically, all living things reproduction is based on what temp and food is available. I mean, there's a lot of... Oh, let me put the link to this paper in the chat, by the way. My bad. I mean, there's a lot of things that affect reproduction. The weather, yeah. Different pressures. I don't know. A lot to think about here. We should have one of these authors join a call one day. I want to get my reading skills up, but I do want to talk to some people that wrote these. Funnily enough, one of the corridors papers we went over once, it was like a college paper, and I do actually know one of the people that was technically on there. I talked to him. He was like, honestly, I wasn't even really that involved with that one. I didn't contribute as much as I should. I was like, no way. The one person I knew was like, I showed up the last day of the group project. No, I'm joking. All right, that was a good paper though. I put the link to that in the chat, and in the Discord I put the link to the Google Drive document that has a bunch of different Daphnia paper things in it. Yeah. Okay, how are we feeling? More Daphnia? Should we do the auction review? The auction review now? Should do another Daphnia thing. I don't know. Let me know what you guys think. I'm gonna look at something real quick. What's up, UEO? I8I, how's it going? What you up to? Daphnia. People say auction. Oh my gosh, bro. Everyone wants an auction. We got the auction heads in the chat. This is crazy. Woo! See, someone cares about Daphnia. I'm with you. I like Daphnia too. I don't know. Water treatment in Daphnia. It's kind of interesting. I'm not gonna lie. Short presentation on reproduction alternatives. What do you suggest? They're like jumping baked beans. That Daphnia. Yeah, I could definitely see that. I had the house to myself, so I'm cleaning worm tubs. Okay. You had the house to yourself, and that's what you're gonna clean the worm tubs? I mean, I can't hate. I do stuff like that when I have the house to myself. I can't really hate. But man, it does sound funny. I didn't know there was Daphnia hate out there. I don't think there's true Daphnia hate out there. Who could hate a cute little Daphnia? It seems like something that would be a little bit unlikely. Okay. I'm trying to see if there's one more Daphnia thing I want to do before doing the auction, but it seems like people want to look at the auction. I get it. Everyone loves auctions. But we're doing one more Daphnia paper. Holy. We're doing one more Daphnia paper. I can't be stopped. I can't be stopped. This is Daphnia Magna as an alternative live fish food for angelfish. Okay? Come on. You can't be too mad about that. You can't be. But do we all feel like we're gaining a bit more of a generalized understanding of Daphnia? I feel like I have. We've looked at some of where they're found. We looked at the life cycle. I feel like I understand that. We've looked at their chemical composition. We have looked at, their defenses against being predated upon, which are kind of pathetic, if I'm being honest, but they're there. Now, this is the keywords we want to see. Council agrees Pollux is overrated. So the abstract says Daphnia magna is a sustainable alternative to Artemia napoli for feeding fish larvae. Artemia have been used as a traditional live food source for juvenile fishes in aquaculture due to their high nutritional value and availability. However, concerns regarding sustainability, costs, and environmental influences of Artemia cultivation have prompted the search for alternative live foods. Daphnia magna, with its inherent nutritional composition, ease of cultivation, and potential for mass production, holds promise as a viable alternative. Though there were slight differences in the adjacent alignment of Daphnia magna and Artemia napoli, the relative examination of data on two individual live feeds showed that at the end of the 21-day experiment, the mean survival rate of juveniles of angelfish was high in Daphnia compared to Artemia as a food source. Survival rates were achieved 85 to 95% in 3 experiments with Daphnia magna as compared to 74 to 79% with Artemia. Get out of here. Get the hell out of here, Aaron. Pack it up. Pack it up. The age of brine shrimp repression is over. I'm decapping all my eggs. They'll never see the light of day. Yo, the Daphnia, the Artemia, it'll never be jumping around no more. It's only the Daphnia now. It's only the Daphnia. I'm sorry to say. I'm sorry to say. Plug my phone in real quick. Okay. In particular, different growth rates were also obtained in 3 different experiments with Daphnia used as a feed. The cost production of Daphnia is much less than that of Artemia. We're reading one paper, and they were saying they were saving like $500 a year just by decapsulating them because they were saving on salt. Okay? Brine shrimp is trash. Garbage. Absolute garbage. We need water fleas. We need water fleas. Listen. Is there a water flea in your life that you've been neglecting? Be honest with yourself. You could be achieving maximum fry survival by embracing the aquatic fleas in your life. People, wake up. It's the 11th of the month. For the past 3 decades, aquaculture has gained importance in different species of shellfish, finfish, blah, 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 broodstock, breeding, food, all the above. This is why it's important that we study this, yada, yada, yada. Let's go. Heck yeah, Daphnia. And these for angelfish, my little fishes can't eat growing ammonia, still too big compared to BBS. Man, that's why you feed the babies. You gotta feed the babies. You guys not getting small like little baby Daphnia for your dad? I just today fed some of the small ones. You gotta get these tiny little fish out of here. My shipment from Greenwater Farms should be here soon. Oh, yeah. Yo, just one more fish with the Greenwater Farms? Come on, add it to your order. JP said, please help me, Daphnia Defense Group. I like this Daphnia defense group thing. This is good. This is real good. Okay. The pure stock of Daphnia Magna. Okay. They used a 250 to 500 micron mesh to collect it. Feeding was done with yeast or chlorella at a cell density of 10 times 106 cells per milliliter. Each gravid Daphnia produces 8 to 10 offspring in about 24 hours. Hell yeah. Dilution of the test tube culture is done daily through several 100 milliliter beakers. The volume is increased to 1 to 2 liter beaker or jars. Feeding is continued in a similar manner in the test tube culture. After 6 days, this culture Daphnia magna are used as inoculums in mass culture tanks. In mass culture tanks, the area is prepared with urea, 10 parts per million, single super phosphate, 25 parts per million, and oil cake ground net, 70 parts per million. After manuring, chlorella was added to the tank. When the chlorella is developed after 5 to 6 days, Daphnia magna is added, 5 to 6 NOS a liter from the stock culture. Daphnia magna multiplies quick, nourishing on phytoplankton. It reaches an ultimate density of 20,000 to 25,000 NOS a liter in 6 to 7 days after immunization. After attaining peak density, it is frequently connected to feed in larval stages. As a consequence of Daphnia magna growth, the concentration in chlorella declines. In sequence, to maintain the optimum density of chlorella, 5% water exchange from the bottom of the tank and additional ground nut oil cake at 70 parts per million is done at an interval of 7 days after the commencement of the first collecting of Daphnia magna. The population of Daphnia magna was recorded using the sedge rafter counter cell, calculated by this formula, where it's T times 1,000 over A times N times V, the volume of concentrate. Yeah, all the math people, you look at this on the screen and understand it, because I can't. You know how I passed my last math class in college? It was like, what is it, algebra and calculus combined or something like that? I'm not even gonna lie. This is how messed up my brain is. Like, I was having such an issue with understanding the steps. I don't know how it worked, but somehow I memorized like the pattern of where the answer was gonna be. So like, I was like, it was way more complicated than if I just followed the steps. I was like, if I take this T and I, multiply it by this number under here, and then I subtract it by this number over here, and then multiply it by this number, I'll get the right answer. And whatever it was I came up with, it was way more complicated than how I was supposed to actually do it. But I just like memorized the pattern. I was like, the answer is always going to be this number here and this number here and this number here. Now, the problem is I don't remember what the hell that was. So if I ever encounter a math problem, I'm totally screwed. I just memorized enough to pass the test. I'll be honest. All right. So they got some angelfish spawned in a hatchery with an overprim injected intramuscularly and a solitary dose of 0.5 milliliters per kilogram fish weight. The growth and endurance rate of the larva was monitored with 2 different diets of Daphnia magna, a common zooplankton, and Artemia. Collection and preparation of the foods. All right, yes, they collected with the net. They got vacuum-packed Artemia cysts, Pro 80 purchased from a local supplier and hatched in the hatchery unit of the Fisheries Laboratory in Midnapore City College. 6 glass aquariums, 24 by 12 by 12, containing 25 liters of water from a borehole supplied with aeration devices were used for the nourishing experiment of the juvenile fish. The tanks were covered with mosquito nets to prevent insect predators from entering. The 6 tanks were assembled into 3 A, B, and C, 2 tanks per group. Each group was stocked with 100 fries and nourished with Daphnia magna and Artemia twice a day for 21 days in the following manner. The research was directed for 21 days with the 2 feeds. The measurements of the weight and total length of juveniles were performed at the beginning and at the end of the research. The number of surviving juveniles in each tank was counted at the end, and the survival rate and specific growth was counted. Here we have the approximate composition of Daphnia magna and Artemia napoli. Math genius Nick using his DDG magnet. What Daphnia defense group magic? Yo, that's crazy. Let's see. Daphnia Defense Group for life. Let's go. We need a t-shirt. I heard even the PhD Aquascaper Bamba supports the Daphnia Defense Group. Oh shit, that's big. We're going somewhere. Daphnia, fairy shrimp are good too. Yep, for sure. I mean, from what I read a long time ago on stream, it seems like they're better nutritionally, apparently, than Artemia. But we'll see if they go over that here. Forget about the mask, though. Put it in the coffin. So Daphnia Magna, we got crude protein at 45 versus Artemia at 42. I'm rounding 45.05. Sorry, 45.06 for Daphnia, 42.44 for Artemia. Crude lipid 18.09 for Daphnia, 7.21 for Artemia. Crude carbohydrate 12.40 for Daphnia, 14.61 for Artemia. Crude ash 14.50 for Daphnia, 9.65 for Artemia. And moisture 89.56 for Daphnia and 72.12 for the Artemia. Okay. And here we have another graph of that same information. So you can see they're not too far apart in a lot of things. The Daphnia have a lot more crude lipid. They have a little bit more crude ash. They have a decent amount more moisture. And we got the table with the different groups, the number of fry and the final number of fry, and the survival rate here. So the highest number, and it's nice, they used 100 individuals, so the percentage is very simple. Like if it's 89%, we know 89 out of 100 survived. Yurio said, I wonder who would win Daphnia versus brine shrimp in a 1v1. I feel like Daphnia had the advantage because the brine shrimp are probably just hatched. So they got a size advantage off the bat. You know what I mean? But I feel like a fully adult brine shrimp would win. Adult brine shrimp look kind of scary. Especially the males because they got those big old... I don't even know what they're called in the front. They hold onto the females with and stuff. An adult brine shrimp would definitely smoke Daphnia any day. They got a pretty big size advantage. So in treatment, in group A1, 89 of 100 fry survived. Group A2, only 74 survived. Group B1, 85 survived. Group B2, 79 survived. Group C1, 95 survived. And Group C2, 76 survived. I'm going to guess all the highest numbers were the ones with Daphnia. And if you're breeding stuff for profit or if you're doing things en masse, I mean, 89 versus 74% is a pretty big difference. 85 versus 79, I mean, that's a little closer. The biggest difference was 95 versus 76. That is a massive difference. Captain said, are they fighting in fresh or salty water? Some of the Daphnia are in saltier water, they said. So how salty? We could also put them against fairy shrimp. I think they'd probably lose even worse against fairy shrimp. There's some pretty crazy fairy shrimp. I ain't gonna lie with you. All right. I'm dropping some frames. I'm not really sure why. All right. Let's see if we can get through this. So survival rates were achieved of 85 to 95% in 3 experiments with Daphnia Magna as food compared to 74 to 79 with Artemia. I mean, they're basically brine shrimp. They're not even worth looking at. Come on. The growth and body weight percentage was recorded higher in Daphnia Magna treatment, 184% to 210% compared to the Artemia treatment, which 170 to 181%. So pretty big. I mean, the low bar of the Daphnia was higher than the Artemia in body weight. So this is graph 2. It's the comparative analysis graph of specific grow rate per day of the angelfish larva fed with Daphnia Magna and Artemia. All the 1 groups are Daphnia and all the 2 groups are the Artemia. And you can see the Daphnia has a significantly higher specific growth rate per day than the Artemia. And then we have a nice graph here of the survival rate, so you can really see how the Daphnia did better there. Now, let's look at the cost. This is fucking crazy. I'm telling you, dog. Any sane person gotta hop on this bandwagon immediately. Okay, hop on the bandwagon immediately. So a total amount of feed purchased and produced by grams. So we got 100 grams of Artemia and 721 grams of Daphnia. The Daphnia cost 0 because they caught them out the damn lake. What is this currency? I'll be honest, I don't know what this currency is off the symbol. I'm uneducated, but it's $2,500 of this currency. Well, not dollars, $2,500 of this currency. The hatching of the cyst has a cost of 250, and then the total cost, 2,750 rupees. Thank you. 2,750 rupees. I believe that makes sense. We'll double check that. Versus the Daphnia, where the total was 322. So the cost difference is pretty crazy. It cost them basically nothing, and the Daphnia outperformed the Artemia significantly. Smart person said that's $30. What, the 2,700 one is $30, and then the other one's about $4. Okay, wow. What is the cost per cyst? I don't. It's still a very big difference. Here we got some figures of the angelfish fry, the Daphnia, them eating. This is kind of like some fever dream stuff. I'm not gonna lie. Like, it's looking a little bit wild right now. Okay, it's something like 80 to 90 Indian rupees to 1 USD. Got it. Okay. The assertion isn't... Okay. So the growth performance at the end of 21 days showed the specific growth rate was slightly higher in Daphnia related to the Artemia. The weight was also slightly better in Daphnia. The growth of fish is believed to be a function of the level of crude protein in the diet, which is higher in Daphnia. This could be qualified to the fact that larval fish may not require more costly imported Artemia feed for better survival rate and growth, because the farmers may easily culture Daphnia magna on a large scale to substitute Artemia napoli to raise newborn fishes. The production cost is also much lower. Also, a lot of things that they study for feeding the Daphnia are like farming byproducts. So a lot of these people, even if the cost might be 0 because what they're feeding them, like the cost of them might be 0 if they go catch them, but then also what they feed them might just be byproducts, so it don't cost them anything. Daphnia is so chunky protein. The assertion is in line with the work of those who stated that the approximate composition of fry feed should consist of 40% protein. Recommended dietary requirement of less than 45% protein for fish larvae is reported that efficient growth of fish depends on feeding the best possible diets at levels not exceeding dietary needs. The moisture content, 89% of Daphnia, is much higher than that of Artemia, 72%. This could mean better digestibility for Daphnia by fish larvae. However, the higher carbohydrate content in Artemia compared to that of Daphnia did not give any extra advantage. The relatively long lifespan of Daphnia magna in water, as observed during the study, coupled with the fact that Daphnia magna is a freshwater zooplankton, makes it much more available and acceptable to freshwater fish in subcontinent environment larvae, which also dwell naturally in freshwater compared with Artemia, which are saltwater. Survival of the larva was higher in Daphnia magna. The general survival rate, however, could be qualified to proper management of larvae and the level of acceptability of the 2 experimental foods by the fish. Daphnia magna is a common food organism applied for the rearing of marine fish larvae. Some have suggested that fish larvae initially have a low endogenous increase in the digestion of food. The result obtained showed that the use of zooplankton Artemia napoli in fish larva culture allowed excellent survival rates. Daphnia magna comprises several digestive enzymes like proteases and peptases, amylase, lipase, and cellulase, which act as exenzymes in the gut of fish. Being larger in size than moina, it serves as a live food for advanced stages of fish. Daphnia magna contains added protein and consequently is a good replacement for Artemia. It's been used extensively utilized as a live food in various hatcheries, etc., etc. Has the advantage of high reproduction rates. Yeah, keep it coming. Let's go. Propaganda. Full propaganda. Let's close this out. Has the advantage of high reproduction rates, wide temperature tolerance, and the ability to thrive on phytoplankton and organic waste. The enrichment of food with Daphnia magna is accomplished with the basis of DHA, which helps make the fish immune to resistance against gill and waterfowling problems. Which helps the fish to make immune resistance again. Yep. The research of the study highlight the both survival and growth rate were higher and more cost effective with Daphnia than Artemia. This results were in accordance with previous findings. Jaffrin et al. 2009 and Reza et al. 2013. Bam. All right. How we doing? How we feeling? Are we still here? Are we still here? If we're still here, we can do the auction review. Can you repeat that? Daphnia Defense Group report this stream. Epic paper, but obviously Daphnia are better. See? Steve, I'm not repeating anything. I don't even care. I don't care. I refuse to repeat anything. Let me just adjust something real quick. Yeah, that's right. That's right. Let's get to this auction fish review. Now, I'll start off. I'll just do the seller's report of what I put in the auction and what I got out of it. Okay. It wasn't the biggest. For some reason, the auction did feel a little bit long at the end. I've just had a very long week. I'm not gonna lie. I was super late. It's kind of a disaster. Yeah. I haven't been streaming a lot because I've been busy, and my life is in shambles. You know what I mean? So it's been hard to get on stream, but here we are. So let me get on the stream my full seller's report for the Aquarium Club of Maryland auction. It was a good auction. A lot of interesting stuff in there. I can see Aaron arguing on the side of brine shrimp in my mind's eye, of course. I'm looking at the Aquarium Club of Maryland auction I went to recently. We're going to check out some of the stuff that was in there and what it went for, and I'm starting with my seller's report of what I put in there. So I only brought 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. I brought 13 lots to the meeting. I didn't bring everything I wanted to. I didn't bap everything I wanted to, but we bapped enough. We bapped enough. So first I bapped 4 crystal red shrimp. Okay. 4 crystal red shrimp that I bred. They went for $22. I couldn't believe it. I couldn't believe 4 of them went for $22. Earlier in the auction, 10 of them went for $35. I was expecting like 12, 15. Nick said, all the articles you had signals you could easily do a 24-hour stream of only Daphnia Magna. Jesus. JP said, you're still maintaining. Just don't go over 4 days. I got you. I got you. I got you. Come on. Let me get through this Valentine's season. Then I had 2 young reticulated hillstream loaches. Those went for 16. And you see the club cut there. So my shrimp went for 22, but I got 15.40. My loaches went for 16. I got 11.20. I had 4 orange laser quarry juveniles that I bapped. Those went for 25. Not too bad. I got $17.50 for that. I had some Christmas moss that went for $6. 2 lots of red root floaters. They both went for $8. So I got $5.60 from both of those. I had some black rose neocaridina coals, like I think 8 of them. Those went for $9, so I got $6.30 from that. 2 more young reticulated hillstream loaches I bred. Those went for $15, so I got $10.50 from that. I bapped 4 infernal tetra fry. That's kind of a little bit of a different, something a little bit different. A rare fish, you might add. The adults are $40 each. Okay, so I actually didn't feel that bad about bapping 4 little small fry. I mean, they're big enough. They're not gonna they're not that small. They're small, but like not that small. You know what I mean? These guys actually started breeding at a pretty small size for me. And I got lucky because I bought 3 adults, and my male killed both females, or I lost both females mysteriously. but 4 fry popped up, 2 of which survived, which ended up being a pair. That pair somehow very quickly started making a bunch more fry, and now I think I'm on my path to making a bunch of those. So kind of a good success story for me. I was able to BAP some of these without feeling like I was gonna fumble them yet again. Aquarium Club of Maryland auction. Yep. Steve said that was a cool fish I never even heard of. Yeah, so this is one of those ones where it's so uncommon, no one knows what it is. Even if the photo looks good, and of course this is a male, like, colored up, they're one of those fish where, like, they can be gray, they can look like this. They're very temperamental. I just saw my male earlier. He looked just like this. It's one of those fish, even if it looks good, if people don't know what it is, they're not really going to want to bid on it a ton. You know what I mean? But they're a fish that they're not commonly in stock. And again, adults are $40 each. So even if you get a bunch of babies, like he paid $15 for 4 babies. Sounds like a great deal to me. You know what I mean? Just a couple months, they'll probably be able to be spawning. And yeah, good luck to Lewis. Anyways, I got $10 from him. Then I bought 4 of my pink Sakura Clarkie crayfish, and those went to Matt, or I know him from Discord as Flashing Boulders. This is a pretty uncommon morph of Procambrus Clarkie that I got. I got a bunch of babies of these. I bat 4 for the points. Luckily, they went to a friend. You see a juvenile male. Aquatic Arts was charging $60 for him. $80 to $90 for the adults. It's expensive. I brought some nice shit to this auction, okay? They went to... I didn't bring much, but I brought some nice shit. So no one can hate on me for that. Those went for $30. I got 21 from that. That went to Matt. He's a good guy. 10 yellow Neocaridina. This one for $25. I got 17. That went to Kim. I bought 4 store-by Corys that I bred. They went to Jacob Water for $22. I got 15 from that. And I bapped a couple Neolamprogus multifasciatus fry. Those went to Brenda for $5. I got $3.50. So all in all, I spent 17. I bought a female longfin bristlenose, super red bristlenose pleco because I want to be able to make longfin pleco babies. I only have male longfins right now somehow. And then I bought $2 for some goldfish pellets, which apparently were opened. But whatever, I'm going to use them for worms or something, so I don't care. But I was like, oh, this is opened already. It was $2, so why not? All right, now let's look at the rest of the auction and what things went for. Now that we've gone through my lot. So overall, I left with $128. There wasn't a lot that I wanted to buy in this auction, and you know, I needed to make it home. Put some gas in the tank. Did Steve get his beta female? Ooh, we're gonna have to find out. So here's a couple of interesting ones. So there's 2 Galaris, 2 females, the Otiri strain. Those went to Cinnamon Girl, I believe. Scotty was selling them. Scotty, why you got 2 lonely Galaris females? Well, they went to Cinnamon Girl for $10, and she got the Galaris male last time. So $10 for 2 females for a Galaris male you got already. Can't complain about that. Can't complain about that at all. $5 for some porthole eye bearers. All right. $9 for some baby rainbow cribs. It's honestly not bad. It's honestly not bad at all. That's what happens when you use a nice photo. Cribs are good looking. Lots of plants. There's a lot of plants. Amica splendens pair plus juveniles, $5. Amica splendens juveniles unsexed. Mick said, what do you think? I just posted a short. Steve, did you already get babies from your betas for that you got them? 3 males, 3... I think this is supposed to be 3 males, 2 females or something. Anyways, some least killifish went for $14. Limia Islay Tiger Limia group went for $5. 2 rainbow tiger Indler pairs went for 10. There's a couple of new people that like guppies coming to the club meeting now. I feel like sometimes the average price of guppies, like some guppies that have been going for nothing recently, like they at least go for like $10 or $14 now, because there's enough people that like guppies coming. A black lace guppy group went for 20. Fancy guppy group went for 9. Trio albino gold platinum molly went for $3. Painted swordtail group as a phosphorus helleride, that went for 18. A full-size mature white worm culture went for 25. If the culture was good enough, that honestly isn't a bad. It looked big, but I didn't get a good look at how many worms. We have one Sabu Saburai Aquatics, putting quite a lot of fancy items in. We had one L387 Tangerine Tiger Pleco, that went for $25. Let me look this up. What the retail price of this is. It's not even like, well, let's see what Saburai sells them for. How about that? A small size is $40. Okay. So, a decent discount. 14K would have been crazy. Yeah, that would have been crazy. A short fin super red bristlenose pleco, probably on the smaller size. I went for 5. One short fin snow white bristlenose. I went for 12. I need to start picking these up because I do like the snow whites, but I kind of want the long fin ones. But 12 for a short fin, I probably would take that too, honestly. But I got nowhere I want to put those right now. We got a common goldfish unsexed juvenile. Someone bought that for $2. A rosy minnow. Someone bought a single rosy minnow. Apparently, it was a donation from the Baltimore County Animal Control or something. All right. Do you have any plecos? I do. I have a couple plecos. Although all my L173Bs are all dying right now, and I don't know why. So, yeah, it's been a pretty terrible month so far as the fish room. I'm losing all sorts of things left and right. Supply and demand. You never know. Yeah. Black worms, plants. Rosy barb trio for 5. Rosy barbs for 7. Dwarf water lettuce for 6. Oh man, CPDs. All right, Steve put some CPDs in. Got $30. 7, 3 and 4, 3/4 inch CPDs. $30. It's not bad. It's not bad. I thought it could have gone to 40. Now this I should have bid on. Siswasser Tang. This big bag of Siswasser Tang. It went to $25. Never mind. You guys got it. I do like Siswasser Tang, but I can wait. 3 Sierra Bulldog Corys. Those went for 18. 3 Orange Venezuelan Corys. Those went for 18. 3 False Bandit Corys. Those went for 12. People weren't too excited about those. 3 Panda Corys went for 14. 1 longfin snow white bristlenose went for 24. It's not too bad, honestly. There was a lot of Siswasser Tang, like 2.5 full. Yeah, it was. I'm not gonna go over my full food list, Mick, but I've had them for almost 2 years now, so it's kind of it's unfortunate. I don't know what's going on. Longfin blue eye lemon bristlenose. That went for 10. A short fin blue eye went for 4. 3 snow white Corys, so it went for 20. They kind of went slow at first, and then all of a sudden people were like, wait a second. Snow white Corys are up? Let's go. I don't know how I really feel about them. If you mix the snow white Corys with normal Corys, like would you get snow whites, a couple snow whites here? Like what would the ratio be? I was kind of curious about that. 3 gold laser Corys. Those went for 15. Damn, I probably should have been on that. 3 the dwarf cuckoo catfish. This is the Synodontis petricola. 3 of them went for $25. 1 long fin super red. That's what I bought. Went for 15. I just opened the bid at 15. I figured that with how people were bidding, I was like, I'll just open at 15. I think I'll probably get it, and I did. Let's go. We got a L306 blood red tiger pleco. That went for $50. Approximately 2 inches. I don't know how much these generally cost. Aqua Imports has them listed for $99, but they're on sale. That's for a medium. So this seems like it was a pretty significant discount on this guy. Scotty bought a yellow bristlenose, he said. Scotty. A yellow bristlenose. Really? Got a Walter worm culture. That went for $14. A micro worm culture for $8. I gotta start bringing some of these. L134 leopard frog pleco. That went for $30. All these foods. We got a vampire crab that went for $10. Some plants. There was a lot of plants, bro. There's almost too many plants sometimes. I'm like, yo, can we get the fish out first at least, so once it's all plants I can dip. Check out. Big bag of Java moss, $16. Green dragon bristlenose pleco juveniles for $14. Grindelworm culture went for $20. Aaron was next to me. He was like, yo, they're bidding up this much? I just like put this culture together. People really want them Grindelworms right now. I'm telling you. Anyone selling Grindelworms, it's your time to make a killing. Everyone's looking for that. There's a bunch of decent sized firemouth juveniles. They went for $14. If I had a place to put them, I was tempted. A triple red pair of cockatoides went for $15, and then another pair of them went for $50. And I believe that I think one of them, it was determined that the bag was actually 2 males, so I think that's why there's a price discrepancy. 4 Epiplades spaghetti juveniles went for $9. Not bad. This was so I didn't realize there was some Hookeraceae moss, but it was a pretty big bag. So when people were bidding up, I was like, really, for the moss? I took another look at the bag. Ooh, it was quite a lot of it. So that went for $40, which honestly it is an expensive moss, so it was a decent amount. Another vampire crab male went for $15. Then we got the Beta Smargina female. Big Steve wanted it. Went for $10. If there had been runners, I 100% would have bumped the lots I wanted it best. Guys said I'm technically ready to drop both Grindles and white worms on my shop. Oh, tempting us. There were 4 Rhinogobius Zoe mixed sex on the gobies. Now, I didn't bid on... I actually probably could fit these in somewhere easily. Here's my thing. Well, one, I just didn't really want them that much. So, like, I'm not going to just buy a fish just because. They went for $30, which ain't too bad. I was... Because I've been having a bunch of losses in the fish room, I'm feeling extra paranoid. I'm like, everything's dying right now. Just everything. I had some issues with temperature. I had some heater struggles. I think things are good now. I got some space heaters. Like, it's warmer in here. I bought some new heaters for the tanks. I feel like really need them. But I don't want to introduce anything crazy disease risk basically right now. Like, I'm feeling very paranoid. And the same seller, LG Felic, I mean, these things happen, but they had this Beta Embellus mixed group, and I noticed in the Beta Embellus mixed group there was a male that was pine coning in there, pretty obviously. And to me, the group wasn't looking too good. So I was like, I don't know what's going on in there. It could be, whatever, freak accident, but I was just like, I'm not touching anything from this seller. Nothing against them. I'm sure everything was probably fine, but I was feeling extra paranoid that day. So I saw one bag that had a pine coning fish, and I was like, nope. And I have a feeling he didn't breed these. So you know what I mean? I was kind of like, I'm going to pass. I'm going to pass this time. Who did get these betas, actually? Cause I'd be curious. Did you get the betas, Big Steve, $40? Because if you did, I'm curious how they're doing. These Boreras Europhthalmoides. Steve said they got cold. I did. Are they got cold after you got them, or you think they got cold beforehand? Or that's what it was. They got cold beforehand. We had these Boreras, the exclamation point Rasbora. What do these sell for? There's like 10 of them. Yeah, like $5 each. They went for $35. Not bad. Not bad. The bidder 16. I had a feeling that was going to be one people were interested in because, you know, the small basically any small nanofish that looks like anything, even a little bit, people are going to be into. The Wabenmuster bristlenose pleco large male. Now, Aaron thinks all catfish are ugly, but on this one, I'm with him. This pleco is hideous. It's actually hideous. Like, I need to be catfished by this catfish in order for me to purchase it. Now, you look up some of these other photos of it. Like, sometimes they don't look like that bad. Like these pictures, it does look nice, but a lot of these are younger pictures of them. You know what I mean? Where the bristles haven't come in. This atrocity, no, I can't do it. Steve said they were cold when they got there. I got them. I kept 3 and gave 3 to Chad. I didn't notice when I split them up. The 3 I have are fine. All right. Well, looks like you lucked out. But definitely, even though I get there late, I do make it a point. Anything I might be interested in, I do go up to the bag. I give them a good look, because stuff like that you're not gonna catch unless you actually take a look at it. So otherwise, I might have been interested. Aphysium bivitanum funge, a pair went for $24. Not too bad. A pair of Elisoma Gilberti plus 2 went for $20. That's a good buying price. Not the worst selling price ever, but like that's, you know, on the lower end. Some Cuberus Panda King went for $25. There's a bunch of isopods. Isopods becoming a fancy thing. You can come to ACM and get some isopods. Not bad. Cuberus Marina Glacier also went for $25. Armadillion Kluge Clown went for $25. $25 is like the buying price for isopods apparently. And then some more Sedontis Petrocoli. 2 of them went for $14. Which is not bad. Now I've been saying that the bristlenose market was Gilberti pair. Yep, Gilberti cost $50 each over here. Yep. Now I've been saying the bristlenose, like they're saturated, but oh my god, there was a lot of bristlenose plecos in this auction, like a lot. Like there's more. We've already seen a bunch, but there's trios of longfin albino plecos, 2 lots of that down here. A pair of red dragon guppies went for $15. Red dragon juveniles for $5. Pair of Steve's Betamacinensis, 1 went for $26. Another pair of them went for $20. I need to put mine in a tank that I think they could breed in. We're getting near the end here. There was a lot of opened foods. A lot of open foods and substrates. Like this is like a partial bag of eco planted. This is like a spirulina flakes. Well, this one's unopened. That's why they bid on up to 10. Some brown gravel. Oh, that one for $1? I should have been on that. Okay, some driftwood went for 13. Saragonite. This Tetra HOB went for, no one bid on it. What? Come on. Sinking shrimp pellets. Yo, I was bidding on the Kin's premium algae wafers, but Val kept bidding them up. I was like, you can have it. It's fine. Excuse me. Steve said those weren't trios. I don't know who sold those as trios, but they were groups of 3. Yeah, well, I think when I saw them, I don't know if it's in the picture, I think I remember saying to Aaron, I was like, how could they have sexed them at the size that it looks like from here? But maybe they got some kind of secret vent examination technique. I think we've gone through most of the fish at this point. We got some bronze cories that went for 15. Oh, here we go. A group of the fasciolatus, the banded barbs. 10 of them went for 20. Oh my god. That's a good price. The gold form Guntherei strain that Aaron made, that went for 30. Not too bad. I think Scotty got those, if I'm not mistaken. I know he at least bid on them. I don't know if he ended up with them. Some zebra isopods, more Kluge. Busan lava rock, that went for 25. That looked pretty nice from Aaron. Trident firm for 18. Gerardinus Metallicus group went for 10. 5 Pseudomagilla furcatus went for 30. Another Pseudomagilla furcatus group went for 35. Peacock gudgeon pair for 25. 10 plus gold bar for 14. 15 Yucatan black mollies went for $15, $1 each. Japan blue double sword group 12. Here we go. The speaker brought some fancy shrimp. 10 orange eye royal blue tiger shrimp went for 70. 6 red galaxy fishbill and caridina went for 40. 10 pure line red line caridina shrimp went for 45. And 10 pure red line caridina shrimp went for 70. Oh, I thought it was 35. It was 45. My bad. I knew this was going to happen. I don't know if I ever brought it up on stream, but I had a feeling the Tangerine Tigers were going to do all right. $60 for 10 Tangerine Tigers. I feel like it's not too bad. Damn, a lot of cribs in here. Oh my god. And Malaysian trumpet snails. They went for $2 this time. Scotty's not making any money off of that. And that is the option. That is the auction review. Okay. Phew. All right. So, listen, we've done the Daphnia thing. I feel like we covered quite a bit of Daphnia stuff. If there's more questions you have about Daphnia that we need to discover, leave a comment. We'll run back on the Daphnia, but I can only read so many hours at a time, right? I'm a dip. I'm gonna get out of here. Okay, I gotta go live. But this has been a nice stream. Back. Hopefully I'll be back again sooner. But yeah, good stream. Getting back into things. Trying to figure out what the next few streams will be about. I want to try some of the higher power heads. That's just something I just want to actually try. So I might do a quick stream at some point, just basically turning them on in a tank, seeing what happens. But yeah, hardest working fish stream. We're still out here. I'm still doing my best. Hope you guys enjoyed, and I'm gonna catch you. I'm gonna catch you on the next one. This has been In My Tanks, hardest working fish stream, Daphnia Defense Group member, and I'll have to catch you guys on the next one. All right, I'm out. Enjoy your evening or your morning. Boop, boop, boop. Wait, I'm still here?